Hi everyone, welcome to Great and Science. We are here to discuss about the molecular basis of life or the DNA. My name is Sir Mike. I'll be your teacher for today. We are already in the week 4 for Great and Science on the third quarter. The content standard for the Science 10 week 4 third quarter is we as a learners demonstrate an understanding of the information stored in the DNA which is being used to make proteins. And the most essential learning competency is the learners explain how protein is made using information from DNA. Going back with our discussion in order for us to understand the concept of DNA, let's go back to the experiment conducted long time ago by none other than Gregor Johann Mendel. In our greater science, we discuss about genetics, on how traits can be passed from one generation to another generation. This process rises up with how do those information can be made and how do those information can be gathered from one generation to another generation in such regards that there is a transformational factor. A series of experiments was conducted in order to prove that DNA is a transformation factor. From Hershey-Chase experiment, Avery, McLeod, and McCarthy experiment, even Graphite experiments gave an idea that DNA itself is a very important thing, and the DNA itself is a factor that contributes on how traits can be passed from one generation to another generation. The evolution of information for the DNA contributed larger to the extent that the DNA itself do not contain just a simple molecule, but it has a what we call parts. And DNA is a polymer, and it has a what we call components known as the nucleotide as its monomer. The DNA nucleotide composed of different parts. The first part of the DNA nucleotide or the nucleotide as a nucleic acid is the nitrogenous bases. Nitrogenous bases is divided into two. We have the pyrimidines and the purines. The pyrimidines and purines are deeper in accordance with their ring structures. Pyrimidines or pyrimidines is composed rather of one single ring, while the purines composed of two chain rings. Pyrimidines are cytosine, thymine, and uracil, while purines are the adenine and guanine. Aside from nitrogenous bases, a nucleotide also composed of a sugar. And this sugar for the DNA is what we call deoxyribose sugar. While the sugar for RNA, another nucleic acid, is a ribose sugar. Deoxyribose because here in our illustration, we will notice that the oxygenation or the removing of oxygen takes place on the ring side of the deoxyribose illustration, while the ribose is just simply both have an oxygen on their part. So again, DNA composed of deoxyribose as their sugar and RNA composed of ribose sugar. So we have nitrogenous bases or nitrogenous bases or bases and sugar. And the third part that contribute to a nucleic acid is what we call the phosphate group. So phosphate group is very important because it connects the sugar backbone to the nitrogenous bases. And those are the parts of the nucleotide or the monomer of the DNA. Let's go with our next discussion, which is what we call directionality. So directionality of the nucleic acid. What is a directionality? So directionality wherein the DNA on what part or what carbon chain does a DNA rather been attached with. Because DNA run into a what we call the anti-parallel structure. Because according to Watson and Crick, DNA is a double helix and they run in anti-parallel structure. This anti-parallel structure compose or composite with how do those following sugars and even phosphate backbone have been attached to a what we call nitrogenous bases, and they run into a what we call 5' to 3' direction and 3' to 5' direction. So primes simply indicate the attachment of a molecule to a particular carbon part. Yeah. So let's continue. So sir, if we have here the 3' to 5' direction and 3' to 5' or 5' to 3' direction, what does it mean? It means that the attachment of the sugar backbone to the phosphate group is in the number of carbon. Take for an example. With this illustration, if we have 5' to 3' direction, meaning to say that the direction of the DNA is going up or up to down direction. From 5', 5 attachment or fifth attachment of the carbon. So from the what we call the bases, 1, 2, 3, 4, and 5, which is, this is what we call the phosphate group. On the other side of our lower part of our illustration, we will notice that it is attached to a 3' group. So what is the 3' group? The end of the bases. So we have 1, 2, and 3. That's why it is called 3' group. And that is the directionality of the DNA, also known as it is anti-parallel. So sir, how do those following bases attached to each other? So there should be what called like a glue on how do those then a bridge on how does or how do rather that the DNA parts or the parts of a nucleotide have been attached to each other. Here's an example. So we have phosphate group, sugar. There is a band between phosphate and sugar group. And that band is very important because it linked the phosphate group to the sugar group. So phosphate to sugar group is being attached by what we call phosphodiester band. While sugar to the nitrogenous bases is being attached by what we call glycosidic band. Or end band. It's sometimes called end band because the end of the nitrogenous bases is N. Thereafter, it's attached with the sugar, and the end part of the nitrogenous bases is being attached. That's why it's something called N-glycosidic band. And the band between the nitrogenous bases to nitrogenous bases. So there is always a 2 hydrogen band between adenine or thymine to adenine, and there is also a 3 hydrogen band between guanine and cytosine or cytosine to guanine. So how do those bands are being informed or being detected? It was when Erwin Chargaff identified the proportionality of those bands. And this is what we call the complementary base rule. This complementary base rule, meaning to say complement, they cannot be broken because it's already a rule. It was according to Erwin Chargaff that adenine is 30.3%, while thymine is 30.3%, and guanine is 19.5%, and cytosine is 19.5%. Okay, this percentage gave rise that there is an equal amount among all organisms. Okay, that resulted to 100%. That makes up an organism. And Erwin Chargaff mentioned that A or adenine always pair up with thymine, pair with cytosine, or thymine always pair with adenine, or cytosine always pair with guanine. And this is a complementary base rule. It cannot be broken because of the experiment that was conducted by Erwin Chargaff, also known as the Chargaff's rule. So which hydrogen bond is strongest among the complementary base pairing? Since adenine and thymine have a what we call and guanine and cytosine, both of them compromise with the bonding system. But since guanine and cytosine have a 3 hydrogen bond, it cannot be easily broken. It has the strongest bond between the bonding system. Okay? Next, the central dogma of molecular biology. This is a very important concept. The central dogma of molecular biology, which happens to all living organism, it takes a general rule on how do the information can be passed from one generation to another generation, such as your hair color, your traits. Most of your traits includes the color of your eyes, your height. This is very important because those traits can be passed from one generation to another generation. And that is indeed by the concentration or by the illustration of the central dogma of molecular biology. So if we have a DNA polymerase, which the DNA undergo a replication process, there are 3 suggestions on how do the DNA replicate. First is the what we call conservative. Second is semi-conservative. And third is dispersive. These 3 proposed model on how do DNA replicate was not being identified before until it was when proved by Watson and Crick that the DNA replicate through the process of semi-conservative manner. So what is a conservative, semi-conservative, and dispersive? This is a very easy way to understand a conservative manner of replication. So the DNA have a what we call double strand. It's a double-stranded. The way how do the DNA replicate or produce another strand is through the process of semi-conservative. If semi-conservative is the manner, means if we have a DNA here, this DNA direct or diverge into 2, forming 2 strands, and these two strands form another DNA. So from one DNA, we can form 2 new DNA. That is semi-conservative. So from one DNA, this one DNA will be unzipped and form another DNA, which is what we call a semi-conservative. If this is conservative, the DNA just replicate and do not form this part. It's conservative. Conservative means the DNA have a single-stranded molecule DNA duplicate itself. Then it's okay, but it's not it is not the real process. And how do the DNA replicate? Dispersive. If the DNA replicate, then the DNA itself will disperse into the series of plasma membrane of the molecule, which is indeed, again, the DNA replicate through a process of semi-conservative manner. Now, after the DNA was being replicated, this DNA will be transcribed, wherein the DNA, a double-stranded molecule, will turn into single-stranded molecule, which is what we call RNA. Okay? So the RNA will form into a mRNA and tRNA. Okay? So the messenger RNA and the transcript or transfer RNA. And it will form into a what we call ribosomes, or form into the ribosomes by making a protein. This schematic process is very important because it leads to a fascinating phenomenon on how do genes have been transformed from one or double-stranded molecule into a single-stranded molecule, which becomes functional in our body. Take a look for this example. If we have this strand of DNA, which composed of C, G, A, and C always form with G, T always form with A, G always form with C, A always form with T. This strand will now replicate. In replication process, or the DNA will replicate, will make their own copies. The DNA will be unzipped by an enzyme known as what we call the helicase. Helicase is an enzyme just like a zipper. The zipper itself, the key for the zipper, it's able to be unzipped. At that molecule that unzipped the zipper is what we call the enzyme helicase, and able to form the DNA is split into two, which is what we call the leading strand and the lagging strand. Leading strand is always formed at 5' to 3' direction, while the lagging strand always add the 3' to 5' direction. So this is what we call the leading strand, and this is what we call the lagging strand. It's simply because it is called lagging strand because the DNA molecule cannot add an usual way of adding a what we call DNA molecules, but instead it uses an Okazaki fragment together with the RNA polymerase in order to add a new sets of DNA molecules. Well, the leading strand continue its process of unzipping the DNA to form its replication part, the part on which the DNA will be replicated normally by the 5' to 3' direction by the DNA polymerase 3. This DNA polymerase 3 adds DNA molecule to the DNA in order to continue the process of the replication. Well, on the lagging strand cannot add the DNA continuously because the leading strand or the DNA polymerase only adds from 5' to 3' direction. But since the direction of the lagging strand is 3' to 5' direction, the DNA polymerase cannot add on the reverse process, but instead it should add first the RNA polymerase, then after that it will be removed by an enzyme, and this enzyme will remove those RNA replaced by the DNA polymerase until the DNA process have been wind up and complete. This process, or the process of this replication, can be sealed up by another enzymes, just like a glue, in order for the DNA not to anneal during the process. And this enzyme is what we call ligase. Enzyme ligase act like a glue to the DNA molecules in order for them to keep intact. And that process of replication indeed takes place to all human organisms and other living organisms on the earth that continue the process of having the DNA replication. Aside from DNA replication, it's very important, of course, not just to understand the concept of replication itself, but the DNA leads to a very prominent picture of constructing its single molecule. This single molecule is what we call RNA. There are 3 RNA in human body: mRNA, the messenger RNA, the transfer RNA, and the rRNA or ribosomal RNA. What are the function of these 3 RNA? They play a great role in the DNA. On what way, Sir Mike? The messenger RNA and tRNA leads to a what we call the transcription of the DNA to the RNA molecules. Well, the ribosomal RNA not transcribe because tRNA acts as transcription material in transcribing the single molecule or the double molecule into single molecule, but the ribosomal RNA serve as an avenue in order to convert the molecule into single strand molecule. In a procedure, we can generally state the process in this way. Okay, let me do it for you. Take for an example. If we have a DNA strand, its a parent strand of A, T, A, C, G, A, T, C, G, T. So this is our parental strand. Okay, this parental strand is very important. It's a DNA template. This DNA template will now replicate. It will undergo a DNA replication. Okay? So, TACG, ATCGT, or thymine, adenine, cytosine, guanine, adenine, thymine, cytosine, guanine, thymine. This whole process, whole template of DNA will go to the DNA replication. So, ito yung process. This is the process wherein we form the leading and the lagging strand. So, T always pair with adenine. Adenine always pair with thymine. Cytosine always pair with guanine. Guanine always pair with cytosine. Adenine always pair with thymine. Thymine always pair with adenine. Cytosine always pair with guanine. Guanine always pair with cytosine, and thymine always pair with adenine. This is the DNA replication. The replicated DNA. Now, on the process of, this is replication. Next process that we will be having is the DNA transcription. So, we will transcribe the DNA template. Okay, we will transcribe again the DNA template. So, this DNA template, the TACGATCGT, TACGATCGT, the DNA template itself will be transcribed. The transcription, we will form, of course, the messenger RNA, the mRNA. So, in messenger RNA, always notice that cytosine or adenine always pair with uracil. So, T to A to U, C to G, G to C, A to U, T to A, C to G, G to C, T to A, and this is A, right? Sorry, this is, it's okay na pala. So, we have AUG, CUA, GCU. What happens, Sir Mike? The A of the DNA template will be transcribed into uracil. And after that, we will transcribe this one. So, A to T, A to U, U to A, G to C, C to G, U to A to U, G to C, C to G, A to U. And this is the mRNA, and this is the tRNA. Okay? Then after that, this concept of forming into three will form what we call amino acids. By looking at the amino acids protein chart. So we have UAC. What is UAC? So we have UAC, that is tyrine, or tyrosine rather. Next, GAU. So we have GA, then U. This is GAU, aspirin or ASP. And we have CGU, which is C, G, then U, arginine or arginine. This is the amino acids. A particular amino acids for the coded DNA template. Okay? In this part, we transcribe the DNA template. And that's the process on how the central dogma of molecular biology takes place from one generation to another generation. Is very visible among all living organisms. We need to know that there are three stop code. Ayan, there are three stop codon. Ang tawag natin sa tatlo, when we divided the letters into three, they are what we call codon. There are three stop codon, and they are the UAA, UAG, UGA. And these three stop codon, meaning to say when they are being formed in the molecule of the transcribed DNA, the transcription of the DNA stop. Okay, the transcription of the DNA to amino acids will stop. or the translation rather. So we have transcription. It is transcription. And we have here, translation. We translate the mRNA to tRNA to the amino acids. And that is our discussion for today. I hope you have something to gain on regarding with how the essential dogma of molecular biology and DNA serve as the basis of life. Thank you for listening and goodbye.